Hi, everyone. Welcome back to day 6. We are now at the second theoretical lecture of the day. And today we're going to talk about how we integrate neuroimaging data, functional magnetic resonance imaging data, and electrophysiology, electroencephalography, with these Bayesian models of learning. Okay, and I'm going to teach the basics. So this is again our agenda for today. My name is Andreea Diaconescu. You've heard from me at the beginning of the day. And we are teaching the data integration part of this modeling session. Okay, so this is the agenda for today. We focus on the first section. So I will give a theoretical overview of dynamic causal modeling for fMRI and for event-related potential data or EEG data. And then Colleen Charlton will present some applications of these types of models. And then at the last, in the last lecture of the afternoon, you're going to hear about extensions of DCM for fMRI for connectomics research, and also how DCM for fMRI can be used to simulate fMRI data. So we've started out with two levels of inference. And if we take into account the Marian levels of inference, which many of you, I'm sure, have heard about, we consider the computational level, which variables or which computations are of particular interest, which computations do we believe underlie learning or belief updating processes during our experiments. And then the algorithmic level has to do with the how. How are these computations, how do these computations arise? How are they evaluated? And we can prescribe different algorithms such as reinforcement learning, hierarchical Bayesian learning, predictive coding. Now we're going to turn to the implementational level. So how are these computations implemented in the brain, and are they in the first place implemented in the brain? How are they implemented through changes in neuronal activity? We can think of it about how they're implemented in relation to neuromodulation. So different ways like that. So we're going to start with the implementational level and looking at different forms of connectivity. So when we think about connectivity, we're talking about 3 different flavors. So we have structural connectivity, the presence or absence of axonal connections. We talk about functional connectivity, which you've heard about on Friday, that has to do with statistical dependencies between regional time series. And then today we're going to spend a little bit more time discussing effective connectivity that deals with causal or directed influences between neuronal populations. And importantly, we are modeling contextual effects of learning or load or following a pharmacological intervention, following, let's say, transcranial magnetic stimulation. So we're really focused on these contextual effects on effective connectivity. Now, very broadly speaking, effective connectivity measures are used to assess functional integration. So how different brain regions communicate with each other to convey a certain cognitive process. And these approaches are based on dynamical systems theory, which means that we care about time delays. So delay is one feature of a connection. And when we talk about effective connectivity, it also means that we are particularly interested in modeling feedback loops, so interactions between, let's say, two regions where the order of that interaction matters. Okay, dynamic causal modeling. Dynamic causal modeling was introduced in 2003 by Carl Friston, Lee Harrison, and Will Penny. And it was initially applied for functional magnetic resonance imaging, or fMRI data. And this paper alone has over 4,000 citations, but DCM itself has been used in thousands of applications. And it actually is growing in popularity thanks to some innovative extensions of the method. So as you've learned on Friday, functional magnetic resonance imaging is a widely employed non-invasive technique that is used to assess brain function. And it has unprecedented spatial resolution, and more recently, it has also unprecedented temporal resolution, so something to also keep in mind for fMRI. Of course, not as great of a resolution as EEG, but there's been a lot of advances in that field as well, in the field of fMRI. However, fMRI signal reflects neuronal activity only indirectly. And that's because of the measurements of the accompanying hemodynamic processes. So an increase in neuronal activity, as you see here on the left, causes localized changes in cerebral metabolic rate of oxygen metabolism. And this, in turn, induces changes in cerebral blood flow, cerebral blood volume. As a result, the oxygen extraction fraction, in other words, the blood oxygen saturation and the concentration of deoxygenated hemoglobin, is changing. And this is reflected in the bold oxygenation level dependent signal, or the bold signal, which we are interested in assessing with fMRI, functional MRI. Now, what is the basic idea of DCM? And as you can see through this diagram, it can be applied both in the context of EEG and fMRI data. The basic idea is that we are dealing with a system where we cannot observe the states that we're particularly interested in. We cannot observe firing rate, changes in synaptic plasticity. But we can only access it in terms of some observed signal, such as bold signal, electromagnetic potential, and so on and so forth. And DCM gives us a formalism where we can describe the hidden dynamics in terms of a series of differential equations that are parameterized and also incorporate some knowledge about the design perturbations of the system. They incorporate this knowledge through the application of inputs. So we have inputs that perturb connectivity in some specific way, but we can incorporate knowledge in terms of priors, in terms of anatomical connectivity between the areas that form our network. To estimate how a neural state translates into a measurement that we can observe, that we can see, such as bold or EEG, we have to invert the model. In other words, we fit the model to the data, we estimate the parameters and reconstruct their posterior distributions. So when it comes to so this is essentially the neural state equation that we can use to translate into a measured signal. And when it comes to fMRI, we have a simple neuronal model, but a very complicated forward model or hemodynamic response model. And on the other hand, with EEG, we have a very complicated neuronal model, but a simple forward model. And we're going to go into that in a moment. So in fMRI, the hemodynamic model is highly nonlinear, and it's based on the balloon model of the bold response that was proposed by Buxton et al. In 1998. And this was an attempt to describe the hemodynamic response as a deterministic function of the dynamic cerebral blood flow change. And it has sort of three components. A model for slow recovery of venous cerebral blood volume after the stimulus, and that's the balloon effect. A model for oxygen extraction fraction based on limited oxygen delivery in the baseline state, and a model for the conversion of dynamic changes in total deoxyhemoglobin and venous cerebral blood flow into cerebral blood volume into the bold response. Whereas for EEG, we are dealing with a complex set of neural state equations that govern the model. The forward model in this case is the lead field matrix. So a lead field is the linear operator that relates brain electrical activity to potentials that we can record on the scalp using EEG electrodes. And the computation of a lead field requires a matrix inversion. And this can be computationally intensive if we had a realistic head geometry that is represented with meshes and a large number of vertices. So basically when we have, for example, information about the subject's own anatomy and electrode positions, we will have a much better forward model, but again, it would be computationally more intensive. So let's take a toy example illustrated in this basic diagram. So we have two regions that are synaptically connected. We have neuron A, two regions, neural populations. Here, let's just reduce it to simple neurons. The connectivity between neuron A in blue to neuron B in green is modulated by a third neuron, neuron C, which is in red. The input enters neuron A, but it does not reach neuron B when neuron C does not fire. So as you can see is that the input affects B only when C is on. When C is off, the input perturbs activity in A, but that perturbed activity in A does not induce a change in B because C is turned off. And this very kind of basic schematic is meant to show that DCM assumes that synaptic strengths are context sensitive, and they depend on spatiotemporal patterns of network activity. And again, I want to stress that in particular with DCM, we are interested in these context-dependent changes in effective connectivity, and less so in the endogenous activity that is kind of happening, the effective connectivity that is taking place in the background. Okay, so this is just to illustrate that in this depiction, C modulates A to B specifically, and the input enters in A. So B has a very low amplitude fluctuation when C is off. Let's consider a previous quite seminal study to convey an example of how DCM works. And basically, this is a task where participants receive the same input, but under two conditions. So they were shown words from the German language, short words, Haus, Dorf, Mond, Baum, etc. And they had to make different judgments based on the conditions. So in condition A, they had to make a letter judgment. So does the word contain the letter A or not? Whereas in the second condition, they had to make a spatial judgment. Is the red letter to the left or to the right from the midline of the word? And this was an elegant task because essentially the network recruitment is determined by the type of condition that the participants are engaged in, whether they make a letter decision or a spatial decision. And this was done by Klaas and Stefan. It was published in Science in 2003. And group analysis showed that there is a distinct pattern of activity in several regions of the brain for letter decisions compared to spatial decisions. So there was this sort of context-dependent recruitment of brain regions. At the same time, there was a distinct pattern for spatial decisions compared to letter decisions. And in particular, this activation pattern involved the bilateral anterior cingulate cortex activation, which was present in both tasks, but showed an asymmetric connectivity or correlation to other regions. So the left ACC was correlated with the left inferior frontal gyrus, and this this connection increased during letter decisions, whereas the right ACC was connected to the right inferior parietal sulcus. This connection was stronger during spatial decisions. So we have the same area, but participating in a different context, in a different network, depending on the type of decisions participants are making. So here, we can see the same effect by looking at the slope. And you see here, signal in left ACC predicts signal in left IFG for letter decisions, but not spatial decisions, whereas signal in right ACC predicts signal in the right inferior parietal sulcus for spatial decisions, but not letter decisions. And this is very nice kind of design for looking at effective connectivity in this network and how the ACC is modulating connections differently based on the type of decision that participants have to do. In this particular study, the authors performed a seed-based functional connectivity analysis. So we don't know the directionality of these connections, whether it's really from left ACC to left IFG or right ACC to right IPS, or whether that involves an additional area C that modulates that connection. In general, there have been many other effective connectivity approaches that sort of extended beyond functional connectivity, such as structural equation modeling or Granger causality. However, the problem with them is that not the problem, but one of the difficulties in interpreting the results of those methods is that they operate at the level of the measurement, at the level of the bolt signal, but not necessarily at the level of the neuronal activation. So, but the mechanisms actually that we're interested in sort of live at the level of the neuronal systems. And the difficulty that we have is that the signals that we measure are being passed through this hemodynamic response function, and they might be expressed differently in different regions, in different subjects. So this will impact the interpretability of, let's say, structural equation modeling or Granger causality in this context, but might be, a reason why one might opt for dynamic causal modeling, such that these differences in the expression of the hemodynamic transfer function or the subject variability expressed at the level of both can be represented in terms of parameter estimates that capture these effects directly. So so we have this kind of dichotomy between the neuronal level and the both level, and of course in DCM they are kind of intertwined. And one thing I want to stress is that in the simplest applications of DCM for tasks like the one that I presented earlier, we're thinking about cognitive networks. And the reason for that is we are interested in that perturbation via context, via cognitive manipulation, not necessarily in these endogenous connections that one might assess using resting state paradigms. Although the method that Peter Bedford will discuss today will look at those applications as well. So essentially the aim of DCM is to estimate parameters at the neuronal level such that the modeled and the measured both signals are maximally similar. And how does it do this? Well, it does this by taking the modeled neuronal dynamics and transforming them into area or region-specific bold signals. Why? Through a hemodynamic model called Lambda. And we're going to kind of go under the hood and try to figure out what Lambda is in order to give us a sense of the cognitive network connectivity at the neural level. So let's take the previous example. So we have this kind of toy network that we're looking at, and let's figure out how DCM is applied in practice. So we have an example in which visual stimuli are presented to the left and the right visual hemifield. So we have a model that incorporates previous knowledge. So stimulation, so one example is that stimulation in a given visual field, visual stimulation, should stimulate the contralateral hemisphere first. And we can incorporate that knowledge through anatomical priors, reciprocal connections present within and between the left and the right hemispheres. Now, the assumption with DCM is that neuronal activity can be summarized by the mean activity in that region. So therefore, each region is represented by a number. So if each activity in each area is represented by one number, then we can compute the derivative, which is a change in activity in time as a linear combination of influences from all the other connected regions. Okay, so this is reflected through the A matrix. This is what I was referring to as changes in endogenous connections or connectivity. Plus, we have the effect of visual stimulation in that area. So we assume that the visual input coming from the respective visual fields is entering, in this case, the left and the right lingual gyri. And the influence of C, the influence of the input U is represented by parameter C, which is the impact of the input on those lower level regions. So we have at the end a set of vector dynamics that change as a function of the temporal course of the input U. And these can be summarized as a matrix where A is the matrix that contains the endogenous connectivity, B, the effects of the contextual manipulation, and C, the input, the external input, the effects of the external input. So again, we are interested in the effects of some manipulation, which we call here context, very broadly, U3, which could be a cognitive task, cognitive load, the effect of a drug, the effect of TMS, etc. We can do this by mathematically augmenting the previous equation by a perturbation, a contextual modulatory term, B here, which is highlighted in this matrix. So B has a specific impact on the connections between region 1 to 2 and between regions 3 and 4. So contextual input affects connections from the right LG to left LG, and then right FG to left FG. Now, let's say the contextual modulation is on and off. If it is on, then we are adding these parameters to the coupling strength. Therefore, the change of state in a system is the effective connectivity to that region plus the effect of the contextual perturbation. And the additional influences are added depending on when the contextual perturbation occurs in time and with respect to the endogenous connection. Okay, so we can summarize this by the following equation, where these ABC are the matrices pertaining to these connections. Okay. So to summarize, so we have these three components: endogenous connectivity, modulation of that connectivity, and direct inputs. So in other words, we model how regions interact given priors governed by their anatomical connectivity. So we have three ingredients that map onto three matrices. A, the endogenous connections between the areas that are present based on anatomical knowledge. B, the contextual modulation and the timing of that. And C, the inputs that are driving the network. So by integrating the equation, you get a time series for X of neuronal activity that you can then pass through a hemodynamic model to generate bold data, and then change the parameters to fit the measured data. So we're going to see this example during the tutorial this afternoon taught by Povlas. Now, what are DCM parameters? So let's recall our calculus. So the integration of a first order linear equation gives you an exponential function. So the coupling parameter A, for example, determines the half-life of X of t. So these coupling parameters are rate constants, and essentially what they do is they tell us something about how quickly an effect is conveyed from one area to another. And they're expressed in hertz, and they are not bounded. So for instance, if the connection from A to B is 0.1 hertz, this means that per unit time, the increase in activity in B corresponds to a 10% change of the activity in A. Okay, so for any DCM that you're working with, the parameters are rate constants and should be thought of or interpreted in this fashion. So let's take another toy example that shows also how contextual input can also affect a self-inhibitory connection. So we also have in DCM temporal scaling of the neuronal responses to the exogenous input. And this is controlled by the intrinsic decay parameter here called sigma. That is shown at the self connection. In particular, sigma controls the rate of exponential growth or decay of the neuronal activity during and after stimulation. And this is a parameter that is enforced to be negative to ensure that the dynamic system remains stable and that the neuronal signal will return to baseline after the end of the stimulus. Now, contextual input or contextual changes can also modulate this self-inhibitory connection. And that can be part of your model. So in this case, we have two regions. The first one is driven by a regularly spaced event trait. Okay, you see here, U1. And U1 enters X1, right? This is the third graph here below. And this activity in X1, you see that it reacts to the input and it decreases exponentially due to the integration of the first order differential equation. Now this connects to the second region here, X2. Now the first region obtains the input first, and the contextual input U2 that we show here that has this kind of sustained effect. This contextual input impacts the self-inhibitory connections, the negative connections for both of these regions. So we see that when U2 is on, this exponential decay speeds up, in particular in the second region that is essentially only affected through the connection from X1 to X2. And it has a self-inhibitory connection that is enhanced, augmented by the contextual input U2. So there's a question in the chat. Just to make sure I understand correctly, the forward model is P measurement given neuronal level, and the inverted model is P neuronal level given the measurement. So we first derive the parameters of the forward model from the data, and again go back to the neuronal signal by inverting the model and feeding the measurements. No, we actually do everything at the same time. So we generate neural activity, we filter it through the hemodynamic response function to get bold, and we adjust the parameters in order to explain the data. But what you're getting at is an important point, because we are actually not interested in the hemodynamic parameters. They are kind of rarely looked at when applying DCM. We are interested actually in the connectivity parameters and the neuronal connectivity parameters. So the hemodynamic response model is parameterized based on previous data. And we have kind of shrinkage priors over parameters, as well as certain parameters are fixed because of known kind of physiology, both physiology, which I will discuss in a moment. But we're optimizing the parameters at once, all of them. Okay, for the single state Dcm, that was single state Dcm that was originally proposed by Friston et al. In 2003. The neuronal activity was modeled using a single state that again summarizes the synaptic activity in a cortical region of a single excitatory neuronal population. However, there's been extensions from there. So the single state neural model was then extended by Mararios et al. In 2008 by adding an inhibitory neuronal subpopulation within each region. This means that this model is actually modeling both excitatory, glutamatergic, and inhibitory GABAergic connections within each cortical region of your network. And this model is represented by two coupled linear differential equations that you see here. And the effect of the neuronal inhibition onto the excitatory population is introduced by negative feedback that is scaled by this parameter mu in this case. And there's been even further extensions beyond that, and this is physiological or adaptive two-state Dcm proposed by Martin Havlicek and colleagues, and this is the work of Kamil Uludag at the University Health Network. So now the physiological Dcm is actually an adaptive two-state Dcm, which is an extension of the Mararios et al. Approach, where the dynamics of the inhibitory state are driven by the excitatory state and scaled with an inhibitory gain factor lambda. And this gain factor allows controlling the relative amplitude of the inhibitory activity with respect to the excitatory activity. So allows basically for an adaptation of this gain. Okay, now how about the hemodynamic form model? And I think often we don't talk about the hemodynamic form model so much because we're not necessarily looking to optimize these hemodynamic response model parameters. But just as we were discussing with the HGF, this is also a response model that kind of links what we're interested in to the observed signal. And just like in the HGF, the response model is often looked at, as a way to map beliefs to decisions, but it's not for many the primary source of interest. The source of interest is actually the learning part. The same thing here, we are interested in the neuronal populations, neuronal activity, but we have to deal with the bold signal. So we have to infer on these more relevant things, but from the signal that that we can measure. So I think it is actually quite important to think about, to understand this hemodynamic forward model, because obviously this will impact greatly how robustly you estimate your effective connectivity parameters and how well you fit the data that you're measuring. So let's start. Basically, the hemodynamic response model incorporates the balloon model, which I alluded to at the beginning of the lecture. So the neuronal model reflects this excitatory inhibitory balance, which evokes hemodynamic changes via the feed-forward neurovascular coupling, and subsequently causes changes in blood inflow. Now, changes in blood inflow are accompanied by changes in blood outflow and changes in blood volume and deoxyhemoglobin content. Now, the rate of blood outflow is influenced by the change in the blood volume. And the change in blood volume and deoxyhemoglobin content is reflected actually in the signal that we measure, the bold response. And so the hemodynamic response model is parameterized in such a way as to essentially describe this mechanism of going from the neural response to the neurovascular coupling and to the hemodynamic model that gives rise to the bold signal. And actually, it's important to note that the hemodynamic model that was incorporated in DCM by Carl Friston in 2003 did actually reflect the existing literature. So basically, says that neural activity creates an inducing signal, ST, that drives blood flow in a linear manner, shown here. So this is the neuronal model, which we've talked about, and number 2 here we have this neurovascular coupling, driving the blood flow in a linear manner. Now, blood flow in the proposal of Friston et al. 2003 responds as a dampened harmonic oscillator. That is, you have this reciprocal feedback effect. So flow changes are caused by the inducing neural signal, and then blood flow in turn changes the inducing neural signal. And this is regulated in the model by 2 parameters, kappa and gamma. And this produces a dampened oscillation at a frequency of about 0.09 hertz that is consistent with the bold signal. So it's consistent also with the Biswal et al. Model. However, there are several studies that came out after that that manipulated blood oxygen and glucose concentrations. And they've shown that oxygen and glucose do not regulate blood flow according to this negative feedback hypothesis. And there's also been hypercapnia studies where people are asked to hold their breath that showed that increased baseline cerebral blood flow only minimally affects stimulus-induced cerebral blood flow changes. So the model by Havicek and Uludag, the physiological Dcm, actually reflects this distinction and this recent evidence related to how the inducing signal affects blood flow in a linear feed-forward only fashion. So neurovascular coupling is regulated exclusively by feed-forward mechanisms. Neurons signal directly to blood vessels via synapses, or they activate astrocytes to release vasoactive agents onto the vessels. And this is reflected in these equations here highlighted in green. So these are the adaptations to the Friston et al. Hemodynamic model. So you have a simple dynamic convolution kernel, which acts as a positively constrained low-pass filter of the neuronal dynamics. And this is regulated by 3 parameters, rho delta, regulated by 2 parameters that I will highlight here, rho and delta, which control the response decay. So when you incorporate feedback, that's a dampened oscillator, but more and more research suggests that this might be strictly feed-forward. And this was applied in a study using retinotopic mapping that has shown that the physiological Dcm model performed significantly better than the standard Dcm, in particular in explaining the empirical data and this undershoot effect of the empirical bold signal that cannot be explained via the standard model due to this feedback loop. Okay, so going back to the question that was raised before. So we're putting it all together in one model. We have a known stimulus function U, that's our task. We have the neuronal state equation that models neural activity according to a series of differential equations. And we have a cascade of hemodynamic differential equations that are used to obtain the bold response. So, so this is the complete model. These are the hidden states, and these are the state equations, the differential equations, and the observation model. The hemodynamic response model also includes a measurement error epsilon, and confounds. Potential confounds can be included in a matrix X, which relates to scanner heating or motion parameters of the participants that are incorporated into the observation model. And essentially, we aim to estimate the model parameters, the theta n's and the theta h's for the neural state model and the hemodynamic model by means of variational expectation maximization under Laplace approximation. So Bayesian inversion, which essentially we change the parameters to maximally explain the observed signal. And this pertains both to the neural state equations and to the hemodynamic response model. So in the result, we have posterior distributions of each parameter that have a Gaussian shape. So Gaussian distribution described by its two moments. So again, similar to the first lecture, we used the Bayesian approach, and the Bayes theorem allows one to formally incorporate this prior knowledge that I mentioned into computing statistical probabilities. Now, in DCM, we're actually using a combination of empirical, principled, and shrinkage priors. So we have empirical priors that are based on anatomical connectivity data and knowledge about, regions, cross-hemispheric connections are bilateral. These are sort of principled choices. But then we also have shrinkage priors. So shrinkage priors essentially we require a lot of evidence. So it's kind of a conservative approach. So we require a lot of evidence to obtain kind of non-zero estimates of effective connectivity. And the posterior probability of the parameters given the data is essentially an optimal combination of prior knowledge and new data. It's exactly the application of Bayes' rule in this case. So, okay, so again, some parameters are principled, such as blood volume is always positive, and the self-inhibitory connection is negative. We have some constraints on hemodynamic parameters that are based on empirical data, empirical measurements. And for the other effective connectivity parameters, they are we use shrinkage priors, where the assumption is that the variance is low and the mean is 0. So we need a lot of evidence to move away from 0. So to be convinced by the data that coupling actually exists. So that's also a way to introduce regularization into your model. So I'm gonna give you a short quiz, and I'll leave it up until you pick up some coffee or have a little break. So essentially, let's say we have an experiment where we have an attention to motion task. So we have dots presented on the screen, and the dots are moving to the left or the right hemifield And in one condition, you have to pay attention to the motion of the dots and respond, whether it's left or right. In another condition, you can ignore them. And so we have a model where we assume that the effect of attention is represented in a particular region of the brain, in this case, this superior parietal lobule. And this activity or this effect of attention represented in the superior parietal lobule affects connectivity from V1 to V5. So the input is driving the motion region of the brain to detect the motion. So this is a quiz that I taken from Hanneke den Ouden, who also taught DCM at previous SPM courses. And I think it's a nice thought experiment for if you are interested to apply DCM to your data. So let's assume basically that you have increased activity in V1. V5 is activated by both the motion and the photic stimulation, V1 only by the photic stimulation, V5 motion and photic stimulation, and superior parietal cortex is only affected by attention. So now you have these 4 different networks. Please identify which one of these 4 different networks would actually explain the changes in regional activity induced by these stimuli in those 3 areas. Okay, go ahead and think about that. So in the meantime, we have a question from Davide. And the question is, I was wondering, how does DCM deal with stationarity assumption of time series, which is a problem for other effective connectivity methodologies such as Granger causality? So exactly, it assumes that it doesn't assume that the signal is stationary. And in fact, because it operates at the level of the neuronal signal or the signal of the neuronal populations, it deals with this issue better than effective connectivity methods like Granger causality that operate at the level of the bold signal. That's one aspect of it. But we have to keep in mind that it is still a simplistic assumption, even in the 2-state model where you have an excitatory and inhibitory populations within each region. And the second question is, have the self connections a correspondence in biology and physiology? So you mean the actual value of the self connections, or just in general having the self inhibitory connection? This effect of synaptic gain, or this modulation in synaptic gain, has basis in physiology. The mean, let's say, or the prior mean for that self-inhibitory parameter is, potentially not. Could be an empirical choice, but it's not necessarily a principal choice, because one could assume that the self-inhibitory connection its effect on gain is driven through the connection with another brain region. But the fact that we have these self-inhibitory effects across all sources, that is based on biology. Okay, so would anyone like to answer this? So let's have a poll. I'm going to use the poll. So we have here upper. So which DCM describes regional activity? Upper left, upper right, lower left, or lower right? Okay. Create poll. Yeah, so please answer that when you have a chance before you see the explanation come up. Okay. Okay, so the lower right is the correct answer. And the thing about the upper left, for example, the reason why that doesn't work is that this connection would not elicit any change in activity in SPC. So you would explain V1 and V5, but not SPC. Now, the second, the upper right, you don't have a driving input of V1. So all of the inputs are affecting the connectivity between V1 and V5. They're not affecting, directly affecting V1, so nothing would happen actually in your network. Then the lower left, you would have the predicted effect in between V1 and V5, so you could explain that, but attention modulating V5 to SPC would not give an increase in activity in SPC as a function of attention, because attention because the effect of attention is not driving SPC, but rather just the connectivity from V5 to SPC. So actually the right connection is, the right network is the lower right. And what this is assuming is that attention is entering SPC and SPC is modulating connectivity from V1 to V5. But perhaps this was a bit of a tricky question, because it's introducing you to a nonlinear version of DCM, which is also possible, where essentially the modulatory factor, the contextual input, is given through another region in your network. Okay, so that's the SPC here. Okay, so we're going to talk about briefly, because I think you've also heard it last year, and actually Colleen will talk about this a bit more in terms of the applications, but essentially DCM has many ingredients and can be used for different data modalities, even fMRIs. And I'm going to talk about EEG today as the more common application. So what DCM for fMRI and DCM for EEG and MEG share in common is one equation that's at the center that describes how relevant states, such as membrane depolarization, evolve over time. The rate of change of X is a function of the current states of the X's, the inputs use, and some unknown parameters, theta. And again, what they share in common is this observation equation, or the forward model, that relates how states evolve through time into a measurable signal that we can record with fMRI, or we can measure from electrodes placed on the scalp. Okay, and in the case of EEG, we have a complex neural model, fast time scales, and a simpler forward model that tells us about how membrane potentials propagate through scalp tissue. So just as for fMRI, the neuronal state equation is coupled with an observation model that gives you the likelihood function. And the likelihood function tells you how likely any data that you might measure given a set of parameters that describe the evolution of the hidden states. So this is, in very simple overview, the roadmap of DCM for EEG, MEG. So we take the characteristics of neurons at the microscale, such as membrane potential or firing rate, and membrane potential to firing rate, sigmoidal transformation, synaptic kinetics, all that is happening at the microscale. We bring that into the mesoscale dynamics of neuronal subpopulations, such as excitatory pyramidal cells and inhibitory interneurons. And you've gotten a bit of the flavor of this on Thursday and Friday from Itai and John's presentations. So this might be kind of an overview of that. But what is sort of the micro to mesoscale passage or path? In the simplest terms of DCM for EEG, we make a mean field assumption. So in other words, each neuron sees all its neighbors, and the average input that it gets is represented by the distribution of the membrane depolarization over that neuronal ensemble. In this case, the theta parameter defines the firing rate threshold, and the area under the curve gives you the proportion of neurons that fire at any time point. Now here, what you see on the right hand side is a Heaviside step function, or a unit step function that is denoted by H. It has the term Heaviside after Oliver Heaviside, and it's a discontinuous function because it has a value of 0 before it reaches threshold, and then a value of 1 after the threshold. Okay, that denotes that is meant to translate a change in the mean depolarization expressed in microvolts to a mean firing rate, action potential. Now the slope of the sigmoid here, S of X in red, reflects the dispersion within that neuronal ensemble. So what we have here is the pre to post synaptic transformation. And of course, if we look at the physiology in great detail, we have very complex things going on. So we have fast kinetics of membrane potentials, different conductances based on the different inotropic, metabotropic receptors. And today I'm not going to talk about conductance-based models, although they exist, but I'm going to focus on simpler convolution models for this lecture. So all of these kind of fast kinetics of membrane potentials that are really interesting and biologically really relevant, they're actually not in the model. So the model sort of tries to summarize this through the introduction of convolution kernels that are separate for excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And the assumption is that these are consequences of these kinetics. And kappa in this case are rate constants. So the convolution of the input to the system, the convolution of the input to the system is performed using this convolution kernels. So a basic example of DCM for EEG, or also event-related potentials in particular, because here we're looking at stimulus-induced perturbations, just like we've been doing with DCM for fMRI. We assume 3 cell populations: spiny stellate cells, pyramidal cells, and inhibitory interneurons. So each parallel equation that is listed here describes essentially the convolution of an input with the synaptic convolution kernel. It's all the same essentially across these different cell types. And this is parameterized with timescale parameters, time to peak, kappa, and decay. The sigmoid function is also parameterized. It has a slope that describes the expected dispersion, and it has an inflection point that reflects the firing rate threshold. So these different cells, different neuronal subpopulations are interconnected with each other based using external lateral connections and extrinsic backward connections and extrinsic forward connections. And this is based on anatomical rules of extrinsic region-to-region connectivity proposed by Feldman and Van Essen in 1991. So, and all of these are modeled for a single source, a single region of interest. So forward connections correspond to afferent pyramidal axons and synapse onto stellate cells, while backward afferents have an impact on pyramidal and inhibitory interneurons outside of this layer 4 corresponding to the stellate cells. There are also canonical microcircuits which provide a refinement to these current models, which were proposed by these current convolution models proposed by Anson and Ritt. And the canonical microcircuit models accommodate the neural sources of forward and backward connections in cortical hierarchies, and they contain distinct, so how they differ from the Anson and Ritt is that they contain distinct superficial and deep pyramidal cell populations that crucially might exhibit different spectral outputs and might be relevant for capturing the signal of interest. Okay, so the way we go from the neural state equations to the observable model, we use this lead field matrix approach, which essentially involves the application of lead field matrix and the performance of a matrix inversion operation. And again, we apply essentially a solution to the inverse problem in this case. So you can consider DCM for EEG as a complex source reconstruction method that has a great deal of biophysical detail on the side of the neuronal state equations that leads to a sort of mapping onto observable signal. But in itself, one can consider it as a more sophisticated source reconstruction technique that is meant to solve the inverse problem. Again, similar to fMRI, we propose prior, likelihood, and posterior. We propose a prior based on anatomical constraints, and we do the exact same thing. So we try to optimize the parameter estimates to maximally capture the observable data. One nuance to highlight when it comes to DCM for fMRI is that here we don't have to look at a specific region of interest. We actually can include all the data that we have from a single subject in order to estimate the parameters of the DCM model. And similar to fMRI, we are interested in contextual changes, in context-dependent changes in effective connectivity. And the same assumption, so DCM parameters are rate constants similar to fMRI, we can interpret them in the same way. Very briefly, you might think about, well, how is there sort of a flow with how one would apply DCM to existing empirical data? And the answer is yes, you would start by applying DCM for each subject, and you would fit the same set of models for each subject. And you would obtain a matrix of parameter estimates that you can then use for a second level analysis or group analysis in order to compare across subjects or to compare across conditions. And this is equivalent to random effects group analysis that can be performed in SPM. And the code that we have provided for this part is actually being applied to fitting single subject data set. Okay, so I'm going to end here and switch gears and invite Colleen up to the screen to give us some interesting applications of DCM for fMRI. Okay, so I see that that most people got the answer right. Lower right is the correct answer. Hi, Colleen.
Hi.
So you can share your screen if you just go at the top, the icon of the folder.
Perfect.
Okay.
Can you see it?
Yeah, we see your the bar with your icon. Okay, now it's gone. There you
awesome.
Go.
All right, great. So I'm just gonna finish this section today, the last 10-15 minutes, talking about some applications of DCM for EEG data. In the next section, they'll talk a bit more about fMRI and its applications. And Andreea, as she's talked about, DCM allows us to look at how the regions we're interested in communicate with each other instead of just conventional statistical analyses, which focus more on regions independently. And today, I'm specifically going to be talking about applications to the mismatch negativity, which we do a lot of work on in our lab. And so some of you may be familiar with it, but just as a brief review. So the MMN is recorded EEG, and it's an event-related potential that occurs when a sequence of standard stimuli are interrupted by the occasional deviant sound. And this can differ in frequency or duration. And this deviant causes this negative waveform, typically between 150 to 200 milliseconds post-stimulus. And so the MMN is calculated by taking the difference between the standard and the deviant ERP, as shown in green. And the MMN can be viewed as a prediction error signal, or signal of surprise. And what's so special about the MMN is that reductions in MMN amplitude have been reliably shown in individuals with schizophrenia, as well as individuals at clinically high risk for psychosis. And so what we can do with DCM is investigate how these different regions communicate during the processing of the standard and deviant stimuli, and extend this to investigate impairments in this prediction error signaling. So just a bit with the MMN, through previous fMRI studies, it's thought that this prediction error signal is reflected in terms of connectivity changes between the primary auditory cortex, secondary auditory cortex, and inferior frontal regions. So basically, our brain tries to infer the causes of its sensory input, and the prefrontal cortex, it's interested in the amount of surprise. So it doesn't care about the difference in the frequency of the stimulus, but rather how different it was from what I previously expected. And so when a deviant is presented, this prediction error information travels up the hierarchy via feed-forward connections. It's used to update our beliefs. And then this prediction about incoming stimuli is predicted back down via feedback connections. And so with DCM, we want to examine this connectivity between the different regions and changes in this connectivity between different clinical groups or under pharmacological influence. So one of the first examples in 2007, looking at the MMN with DCM, the authors focused on the role of backward connections to investigate the ERP elicited by the deviant stimulus. So based on previous literature, the authors assumed these 5 sources, and they compared 2 models, both with the same source architecture, but just varied with the presence of backward connections. And so it's important to note DCM, it's hypothesis-driven method. So it doesn't explore all possible models, but instead it tests these specific mechanistic hypotheses and then defined in terms of these specific connectivity models. And what the authors found, so fitting DCM to the EEG data from 11 healthy participants and zooming in at 180 to 260 milliseconds post-stimulus, so it's around the time of the MMN. And so the best given model is the one with the highest log evidence. And the graph on the right here shows the difference in log evidence between the forward-backward model and forward model. So the points outside the gray zone imply very strong confidence that one model is more likely. And the negative points are in favor of the forward model, and the positive points are in favor of the forward-backward model. So what's interesting is that during this earlier component, the forward model is favored. So these forward connections are sufficient to explain the ERP generation. But in the later time component, it's the forward-backward model that's favored. And so these backward connections become more essential. And this is likely around the time where the activity returning from these higher cortical areas is passed down the hierarchy. And to just kind of drive the point home, using both models, the authors simulated deviant ERP responses, shown in the solid black line, and then compared this with the real or observed data shown with the dashed line. And we can see that in the beginning, the both models provide a pretty good fit for this earlier response, but it's the forward-backward model at this later component that is the best fit. So again, these findings are just in line with the hypothesis that the brain is continually trying to infer on incoming sensory input, which entails the passing of these prediction errors up the hierarchy, and then later on passing the predictions about incoming stimuli back down via these backward connections. And a second example done about a year later, here the authors are investigating the different networks involved in the MMN. So in the first example, they assumed the 5 regions involved, which again was based on previous literature. But here what they're doing is we can see with the different model space on the right here, comparing different models. And so this addresses the hierarchical network ranging from 1 to 3 levels. And in the top here, these models are looking just at changes in extrinsic connections alone, so these forward and backward connections. But in the bottom 3 models, the extrinsic connections are in combination with the changes in intrinsic connections at the level of the A1. So model S5I here kind of covers the hypothesis that both local adaptation within the primary auditory cortex and interactions within this temporal frontal network underlie the generation of the MMN. And what the authors found was that both the standard and deviant ERPs were explained best by the same model, S5I. And just zooming in on the winning model, the authors performed a post-hoc t-test on the B matrix, which Andreea talked about before. So these are the connectivity parameters between the regions, and basically found that the MMN could be explained by changes in the strength of these connectivity parameters, both between and within these cortical sources underlying the network. And so just to recap, we showed that DCM can model this MMN prediction error signal, and it's reflected in terms of changes in extrinsic connectivity between the primary auditory, secondary auditory, and inferior frontal regions, as well as local adaptation within the A1. And just to show how we would model it, Andreea talked about the different matrices before, but so DCM allows, it provides this matrix format which we can input our connections. So the A matrix, as mentioned, defines the baseline connectivity between the regions. So for MMN generation, we assume we have feed forward, feed back, and these lateral connections. And then the B matrix, they captures whatever's affected by our task. So whatever connectivity changes are driven by the manipulation. And here we argue that the cognitive effect of this stimuli, the deviant stimuli, is modeled by both feed-forward and feedback connections, shown in red. So how this would look, again, we have our 5 regions, and you can think of the columns reflecting from, and rows are to, so it's the region-to-region connectivity. So from the left A1 to the left STG, is seen here, we believe a connection, and from right A1 to right STG. And then in our matrix, we also define our backward connections. So same idea with from left STG to left A1, from right STG to right A1, shown here. And we also have our lateral connections, which just makes sense anatomically. We know these regions are interconnected. So from right A1 to left A1, and the reverse from left A1 to right A1. And then it's with our B matrix that we want to model the connections we believe are modulated by our experiment. So again, these are the ones shown in red here. And just to kind of show how this would look in Matlab, at the top here, we just have our source name, so left A1, right A1, etc., as well as the MNI coordinates for where these are. Got our A matrices here, so our forward connections, backward connections, and lateral connections defined with the ones. We have our B matrix, again with the ones defining these connections we believe are modulated, and our C matrix. And this represents, as Andreea talked about, the input entering our system. So it's just a vector, and it's the same number of elements as the number of regions in our model. So in our case, it's 5. And we are assuming the input enters the primary auditory cortex, the lowest level region. And what we'll get from DCM are these connectivity parameters of the B matrix estimated for each subject. And then we can examine these using different statistical analyses. So just to kind of show this, this example looked at, or used the MMN and DCM to investigate group differences between healthy controls, individuals in a vegetative state and minimally conscious state. So the authors wanted to see if the MMN could differentiate between the three. And on the top, we have the 11 different models that the author specified, which just differ in the connections and areas involved. And it was found that model 11, so the one with the 5 areas and forward and backward connections, best explain the data. So then using these connectivity parameters, the authors looked at group differences. And so the only significant effect, and these are the parameters, sorry, from the best fitting model, so that model 11. And yeah, so the only significant effect was on this backward connection between the right IFG and right STG. So here the backward connection was significantly impaired in vegetative state patients compared with controls. And the minimally conscious patients showed this preserved top-down connectivity compared to vegetative state. It wasn't significantly different from controls, but there was a difference between these two. So the authors basically showed that the selective disruption of this top-down processing from this higher level of the cortical hierarchy can potentially lead to a loss of consciousness in brain damaged patients. It can be used also to clearly differentiate between the vegetative state and minimally conscious state. And then just one last example looking at pharmacological interventions. So as I mentioned in the beginning, the MMN has been reliably shown to be attenuated in people with individuals with schizophrenia. And similar MMN reductions can be achieved with the NMDAR antagonist ketamine. And ketamine also induces psychosis-like experiences in healthy individuals. So in this study, the authors investigated 8 different models between the placebo and ketamine condition that underlie the generation of the deviant response. And so here the red represents the different connections of the 8 different models. So M1, for example, reflects no modulation of connections. M2, just forward connections. And then like these bottom models here, M5 to 8, they also incorporate local adaptation in the A1, similar to some of the previous studies. And it was found for both placebo group and ketamine group that model 8 was the best model. And then again, using model 8, the authors looked at the connectivity parameter estimates, comparing the placebo and ketamine conditions. And it was found that plasticity in this forward connection, so from the left A1 to left STG, showed a significant change under ketamine. And a similar trend was observed in the homologous right connection, but it didn't quite reach significance. And there was also a significant correlation found between ketamine's reduction in this coupling and self-reported reductions in control and cognition ratings. So this is under the influence of ketamine. And these ratings are reminiscent of schizophrenia symptoms. So a greater reduction in control and cognition was associated with decreased coupling in the left A1 and left STG. And so just to finish off, kind of as Andrea mentioned, there is an extra homework assignment, and there is more info on GitHub, and that's where it can be found. And again, in the day 6 folder and under the EEG folder, you'll find this MMN master script here. And it will take you through the pre-processing and the model-based analysis using the HGF of one subject's EEG data. And at the very end, you can also model this using DCM and explore the different parameters, attempt to answer these questions. And yeah, if you have any questions, probably went a little over time, but we'll be around after the next section.
Thank you so much, Colleen. So there was a question, I answered it in the comment. So just to repeat it for the rest, you showed this decreased feedback from IFG to STG in the study by Garita et al. 2008. And the question was whether this decreased feedback is counterintuitive. And I just wanted to highlight that what was modeled there was the effect of the deviant, so which one assumes to be the effect of a surprise event that violates a statistical regularity. So this could be thought of as reflecting the impact of an incoming prediction error. And this is expressed through increased connectivity from A1 to STG and not from IFG to STG. However, the prediction, like you mentioned, might be conveyed from the top-down regions or from the IFG to STG, so that as the deviant is repeated and becomes a standard, then that connection from the inferior frontal gyrus to the STG is increased. Any other questions? So maybe one clarification. So the pharmacological ketamine effects are, very important to think about. So in the example 4 that you showed, Colleen, so we were quite surprised that the effect of ketamine on the manner in which ketamine modulated effective connectivity was most robustly expressed in these sort of bottom-up connections from the A1 to the STG. And this was surprising because we had expected that this would be reflected in the top-down predictions. But it turns out when we integrated the hierarchical Gaussian filter in order to differentiate between outcome-related and volatility-related prediction errors, we were able to find that actually this reduction in bottom-up connectivity is conveyed through a reduction in the volatility prediction error expression. But of course, when you perform a standard analysis and categorize the deviant and the standard as two separate conditions based on the experimental design, you cannot really distinguish between these two types of prediction errors. So that's something important to keep in mind. And when you try out the homework using the code, you will be able to do exactly the same. You'll be able to integrate the HGF that was covered by Daniel in the previous tutorial with these DCM for EEG models. So if there are no questions, I invite you to join us to GatherTown, and thank you for your attention. Bye.